Welcome to the class 11 students. I will be continuing with the chapter cell division. This is the part 2 of that chapter, and in the previous video, that is the part 1 video, I have discussed about the types of cell division and cell cycle and the phases of cell cycle. Now today I will be talking about cancer and what are cancer cells and what is malignancy. Okay, so to begin with, cancer can be defined as a disease involving heritable defects in cellular control mechanisms, resulting in the formation of malignant and usually invasive tumors. Now, to explain this definition, it is a heritable involving heritable defects, means those defects that have been heritable from the previous generation, and these defects are mainly in the cellular control, means how the cells are being controlled. Means if the cells are not properly controlled, they will be means injured or wounded or abnormal cells, and then they will become malignant, means then they will become cancerous and usually invasive in nature, because they will invade the other body parts and will invade the other cells also. Okay, so here you can see two types of tumor, that is benign tumor and malignant tumor. But before that, let me tell you what is tumor. The rapidly dividing cells that accumulate in a certain part of the body, forming a lump or a growth like structure, is known as tumor. So a tumor is not very dangerous, it is not invasive, it is just a rapid lump which is formed of rapidly dividing cells. It can form at any part of the body. Okay, now there are two types of tumor: benign tumor and malignant tumor. Firstly, benign tumor is non-invasive, means it is not really harmful, it is not dangerous. Here the uncontrolled cell division stops after a certain point of time, and things do not get really out of the hand. Okay, they are still under control. But when benign tumor becomes malignant tumor, it becomes invasive and it becomes cancerous when the cell division means the dividing cells migrate to other parts of the body and form secondary tumors there and affect the cells there as well. So benign tumor, these cells are not cancerous and will not spread to other parts of the body. But in malignant tumor, you can see that the cells are spreading from their original location and they are now cancerous. That is why they are known as malignant tumors. Okay, now let's talk about how these cancer cells or malignant cells are different from normal cells. Okay, so there are a number of differences between normal cells and cancerous cells. Firstly, here we can see a few differences. Mainly the cancer cells are large, variably shaped. They have large variably shaped nuclei. So if you can see in the normal cell, the blue very small circular structure is the nucleus, but in case of the cancer cell, the nucleus is very large and they do not have a proper shape. Okay, next, moving on to the next difference is that they have many dividing cells, disorganized arrangement. So no proper arrangement of cells are there. The cells, since the cancer cells are formed as a result of uncontrolled or rapid cell division, so there is no arrangement of the cells. As a result, the cells are disorganized. Okay, next, the cells itself, they have quite variability in size and shape, which should not be the case in normal cells, because the same type of cells, they should have the same size and shape. Lastly, loss of normal features of the cell, and mainly malignancy is also a very important characteristic of malignant cells. When the cells lose the control over cell division and they then spread to other parts of the body. Other characteristics include loss of adhesion, means malignant cells show less adhesion to the support, or they show great increased mobility, that is they move around a lot. So students, now I will talk about the different stages of mitosis. Previously I have told you that mitosis is divided into two phases which are known as karyokinesis and cytokinesis. So here you can see that the cytokinesis is already written here, and the first four phases belong to the first or the initial phase of mitosis, which is known as karyokinesis. So okay, so karyokinesis is the division of nucleus which occurs through these four phases: prophase, metaphase, anaphase, and telophase. And then after the nucleus is divided into two separate nuclei, cytokinesis takes place, which is the division of cytoplasm. Now we will talk about each of these phases in details. Firstly, talking about prophase. Now the word pro means before, or primitive, or initial. Okay, this phase of karyokinesis, what happens in this phase is mainly the nuclear membrane and the nucleolus begin to disappear. So if you can see in this picture that this is the nuclear membrane, and as you can see that there are discontinuations in the nuclear membrane, which suggests that the nuclear membrane starts to disappear or disintegrate, and the chromatin fibers become tightly coiled. So here these chromosomes, which are also known as chromatin fibers, they become deeply or tightly coiled and more condensed, and they form these structures which are known as chromosomes. Okay, so mainly two events happen during prophase, that is the nuclear membrane starts to disappear and the chromatin fibers become more tightly coiled and condensed and form these structures which are known as chromosomes. Okay, so now the first stage of mitosis, or the first stage of karyokinesis of mitosis, we have discussed that, and that is prophase. Okay, now we will now you will have to learn the characteristics of prophase. The two main characteristics I have already discussed here, and rest characteristics or in details you will find in the book, but these two are the main and most important characteristics. Okay, moving on to the next phase, or the second phase, which is the metaphase. Now what happens during metaphase? This phase of karyokinesis is when the chromosomes have become aligned on the equatorial region. So if these are the two poles, this is the equator of this cell, and as you can see that the chromosomes have arranged themselves near the equatorial region, and the centromeres, if you can see all the centromeres here and here, they lie along the spindle fibers. So these are the spindle fibers. You have learned about spindle fibers previously while we were discussing the previous chapter. So all the spindle fibers which radiate from the two different poles, they connect with the centromeres of each chromosome, and metaphase is the second phase of karyokinesis, and it is when the nuclear membrane is completely destroyed. You cannot see here the remains of the nuclear membrane can be seen, but here nothing can be seen. Okay, so spindle fiber has also developed in this stage. Centromeres of chromosomes lie at the metaphase plate. So these are the certain or the main characteristics of metaphase. Moving on to the third phase of karyokinesis, which is anaphase. Okay, so in this phase of mitotic cell division, the two chromatids of each chromosome are separated. As you can see in this picture, the two chromatids have separated and they go towards opposite poles. Okay, so this is the shortest phase of out of all the different phases of karyokinesis of mitosis, and during this phase what mainly happens is that the chromatids, the sister chromatids, they separate from each other. The centromere no longer binds them, and each of them move towards opposite poles. Now each chromatid is now a full-fledged chromosome, and chromatids are attracted to the poles at kinetochore by kinetochore spindle fibers. Okay, so these are the different characteristics of anaphase that you will have to learn, but the main phase, or the main feature of this phase is that the sister chromatids separate and they move towards opposite poles. Okay, so I will continue with the next two phases, or the next phase of karyokinesis, and then the last phase of mitosis, that is cytokinesis. So these two phases I will continue in the next video. Till then, I hope you have understood till this part. Thank you.